Sandwich ELISA. Unlike direct and indirect ELISA, the sandwich ELISA begins with a capture antibody coated onto the walls of the plate. It is termed as sandwich because the antigens are sandwiched between two layers of antibodies, capture and detection antibodies. After adding the capture antibody to the plates are then covered and incubated overnight at 4 degrees Celsius. Once the coated step is complete, the plates are washed with PBS, then buffered or blocked with BSA. The buffer washes are carried out for at least 1 to 2 hours at room temperature. Finally, the plate is washed with PBS once again before the addition of the antigen. The antigen of interest is then added to the plates to bind to the capture antibody and incubated for 90 minutes at 37 degrees Celsius. The plate is re-washed and the primary detection antibody is then added to the plate and incubated for another 1 to 2 hours at room temperature, followed by a buffer wash. Then a secondary enzyme conjugated antibody is added and incubated for another 1 to 2 hours. The plate is re-washed and the substrate is added to produce a color change. The sandwich ELISA has the highest sensitivity among all the ELISA types. The major disadvantages of this type of ELISA are the time and expense and the necessary use of matched pairs, divalent or multivalent antigen, and secondary antibodies. Here is the diagram of sandwich ELISA. Now the fourth one is the last, competitive ELISA. The competitive ELISA tests for the presence of an antibody specific for antigens in the test serum. This type of ELISA utilizes two specific antibodies, an enzyme conjugated antibody and another antibody present in the test serum if the serum is positive. Combining the two antibodies into the wells will allow for a competition for binding to antigen. The presence of a color change means that the test is negative because enzyme conjugated antibody bound the antigens, not the antibodies of the test serum. The absence of color indicates a positive test and the presence of antibodies in the test serum. The competitive ELISA has a low specificity and cannot be used in dilute samples. However, the benefits are that there is a less sample purification needed. It can measure a large range of antigens in a given sample, can be used for small antigens, and has low variability. Here is a picture of competitive ELISA which completed in 4 steps. The 4 types: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Now the latest research and advancements in ELISA reader and its application. In diagnostic, the first one, diagnostic test. Enzyme-linked immunosorbent assay are applied in many diagnostic tests. Some of the uses of ELISA can include the following: detect and measure the presence of antibodies in the blood, autoantibodies, antibodies against infectious diseases, hepatitis A, B, C, HIV, etc. In biomedical research, tracking disease outbreaks, cholera, HIV, influenza, detecting past exposures, HIV, Lyme disease, hepatitis. In drug discovery, detecting drug abuse, amphetamine, methamphetamine, 3,4-methylene dioxymethamphetamine, cocaine, benzoyl The material from references, links. You can also watch this procedure, complete procedure, from YouTube by this link. These are another links. The available companies of ELISA reader are Biored, Abcam, and Siemens. The tube size used in wells ranges from 18, 25, and 15 microliters. The software used in ELISA reader is typically a software package such as the ELISA reader for Windows or the ELISA reader for Mac. Thank you.